Deep inside the mesophyll cells of a plant leaf operates a network of highly adaptable biological factories. These are the plastids, a diverse group of organelles that manage everything from synthesizing the plant's food to stockpiling its essential nutrients. Before any of these cellular factories take their specialized form, they all begin as the exact same structure, the proplastid. This is a tiny, undifferentiated blank slate. Depending on the tissue type and environmental cues like light exposure, the proplastid develops specialized internal machinery to meet the cell's immediate needs. This hierarchy flowchart maps out the 3 primary developmental branches a proplastid can take. If a leaf cell needs energy, it develops into a green chloroplast. If a flower needs to attract pollinators, it matures into a pigment-rich chromoplast. And if a root needs to hoard resources, it becomes a colorless storage unit called a leucoplast. Within that storage category, the specialization goes even deeper. A leucoplast will differentiate into an amyloplast to pack away carbohydrates as starch, and ileoplast to store energy-dense oils and fats, or an alluroplast to accumulate proteins. Capturing solar radiation requires completely different biochemical machinery than crystallizing fat-soluble pigments or stockpiling starch granules. Yet because they all mature from the same proplastid ancestor, they share a unified underlying architecture. They are a single, modular toolkit that ensures the plant can survive and propagate. Looking at this 3D cross-section of a chloroplast, we can see the first major feature of that shared architecture, a double membrane defense system. The outer membrane contains specialized pores, making it highly permeable to small molecules. Just beneath it, the inner membrane acts as a highly selective gatekeeper, tightly regulating exactly which metabolites can enter or exit the interior. Past that inner gate is the stroma, a semi-fluid matrix where carbon fixation occurs. Suspended within this fluid are structures arranged like stacks of coins. An entire stack is called a granum, and each individual flattened sac within that stack is a thylakoid. This is where the light-dependent reactions of photosynthesis actually happen. These stacks are not isolated. They are physically connected by horizontal membranous bridges called stroma lamellae. This continuous, highly folded network packs a massive amount of surface area into a microscopic volume, maximizing the density of light-harvesting pigments exposed to the sun. But a detailed chemical analysis of the stroma reveals a biological anomaly. Floating freely in the fluid alongside the stacks are distinct circular DNA molecules and small 70S ribosomes. In the rest of the biological world, these specific genetic structures are found in one place, free-living bacteria. The presence of bacterial genetic machinery inside a plant's solar factory means the chloroplast is a surviving remnant of an independent organism that was integrated into the host cell. In the latter half of the 20th century, biologist Lynn Margulis championed the endosymbiotic theory to explain how this bacterial machinery entered modern plants. Over 1 billion years ago, a massive predatory eukaryotic host cell engulfed a free-living photosynthetic cyanobacterium. But instead of digesting the bacteria for fuel, the host cell retained it. As the bacteria was pulled inside, the host's engulfing membrane wrapped entirely around the bacteria's original plasma membrane. This specific physical interaction created the signature double membrane envelope that encloses all primary plastids today. Inside the host, the trapped cyanobacterium enjoyed a stable environment and physical protection. In exchange, over millions of years of coevolution, the bacteria transferred the vast majority of its own genetic code to the host cell's nucleus, cementing an obligate, permanent partnership. Integrating photosynthesis into eukaryotic cells enabled the colonization of land and the eventual oxygenation of Earth's atmosphere. The biological integration did not stop with a single engulfment. The process repeated itself, creating a microscopic Russian nesting doll scenario where the entire resulting photosynthetic cell was consumed again. This evolutionary tree maps how diverse lineages acquired plastids. Heterotrophic hosts like euglenoids and chromovalulates engulfed entire algae cells containing primary plastids. By wrapping its own membrane around an algae cell, the structural math compounds, creating complex plastids with 3 or 4 distinct bounding membranes. Once engulfed, the secondary host gradually dismantled the unnecessary parts of the trapped algae. The algae's original nucleus was completely discarded or reduced to a tiny remnant called a nucleomorph, effectively stripping the captive cell down until only its plastid remained. Secondary endosymbiosis allowed the biochemical machinery of photosynthesis to jump laterally across completely different branches of the tree of life. This mechanism rapidly created diverse autotrophic lineages that now form the foundation of global marine ecosystems. This summary grid aligns plastids with their functions. Chloroplasts drive energy conversion, chromoplasts facilitate signaling through color, and leucoplasts store nutrients. Every variation traces back to a prokaryotic endosymbiont, their capabilities resulting from millions of years of structural reduction and genetic coordination with host cells. Whether observing the green canopy of a forest, the red coloration of a tomato, or the starch stored in a potato, we are witnessing the biological legacy of an ancient endosymbiotic relationship that still powers the modern biosphere.